Hello students, so this is our unit Plant Physiology and chapter is Respiration in Plants. Today we'll study about electron transport system and oxidative phosphorylation. In electron transport system, we'll study that this is the metabolic pathway through which the electrons pass from one carrier to another. So this is commonly called as ETS. So let's have a brief study of electron transport system and oxidative phosphorylation. So the metabolic pathway through which the electron passes from one carrier to another is called electron transport system. It is present in the inner mitochondrial membrane. You can see in the diagram, ETS comprises of the following complexes. Complex 1 is called NADH dehydrogenase. Complex 2 is succinate dehydrogenase. Complex 3 is cytochrome BC1 and complex 4 is cytochrome A2A3, cytochrome C oxidase and complex 5 is ATP synthetase. So NADH produced in the citric acid cycle is oxidized by NADH dehydrogenase. Electrons are then transferred to ubiquinone located in the inner membrane. It is a protein which is present in the inner membrane. So FADH2 is oxidized by succinate dehydrogenase and transferred the electrons to ubiquinone. So this is in the continuation with the previous slide, electron transport system. This is the third step, the reduced ubiquinone. Ubiquinol is also called. It will then oxidized with the transfer of electrons to cytochrome C via cytochrome BC1 complex. This is called complex 3. Now cytochrome C is small protein attached to the outer surface of the inner membrane and acts as a mobile carrier for transferring electrons from complex 3 and complex 4. When electrons are transferred from one carrier to another via complex 1 to 4 in the electron transport chain, they are coupled to ATP synthetase, that is complex 5, for the synthesis of ATP from ADP and one pyrophosphate molecule. One molecule of NADH2 gives rise to 3 ATP. One molecule of FADH2 gives rise to 2 ATP. Oxygen plays a vital role in removing electrons and hydrogen ion finally helps in the production of water. Phosphorylation in the presence of oxygen is called oxidative phosphorylation. Now we all have studied about the mechanism of membrane-linked ATP synthesis as explained by chemosmotic hypothesis in the earlier chapter, that is in the photosynthesis. So the energy released during the electron transport system is utilized in synthesizing ATP with the help of ATP synthetase enzyme, that is called complex 4. Now we'll study about this complex. This complex consists of two major components, F1 and F0. F1 is headpiece, is a peripheral membrane protein complex and contains the site for synthesis of ATP from ADP and pyrophosphate. F0 is an integral membrane protein complex that forms the channel through which protons cross the inner membrane. The passage of protons through the channel is coupled to the catalytic site of the F1 component for the production of ATP. For each ATP produced, two protons pass through F0 from the intermembrane space to the matrix down the electrochemical proton gradient. Means for the production of one ATP, two protons are consumed.